Hey guys, so thank you for coming to talk by UBIC. And then so for today's talk, we've introduced Dr. Lukas Chavez from UCSD School of Medicine, so that his talk will be about chromatin states and confirmation. Please give him some applause.
I also have some notes on paper, old school. Thanks a lot for the invitation. I think that is really a very good concept. It's very challenging for me. I'm not used to use the chalk at all anymore, but I think that's the idea of it. I mean, I haven't done a lot of teaching yet, and I haven't taught for undergraduate. So it was really a preparation for today, was somehow a challenge to see how I take my talks that I typically present at scientific conferences and make it suitable for this audience. I thought, because I'm still relatively new to UC San Diego, and it might also be a good idea if I introduce myself. I actually grew up in Germany, and this is why you have to deal with my German accent. Shortly after high school, I started my bachelor studies with a major in biology. And it was after my first year that I realized that the genomic revolution is about to shift the field of biology just more and more towards data science. And this is why I actually changed my major. I had to change the universities within Germany, and I started to study bioinformatics. The German education system is slightly different to the US system. I graduated with a bachelor's in bioinformatics, and a few years later I received my master's degree in bioinformatics, what was pretty new at that time. And afterwards I enrolled into a PhD graduate program at the Max Planck Institute for Molecular Genetics and at the Free University in Berlin. And it was during that time that I became interested in epigenetics and chromatin biology. And as a PhD student, I was co-supervised by two group leaders. One group leader was more on the experimental side. He was culturing human embryonic stem cells and was differentiating them along the endodermal lineage to derive more differentiated human cell types like hepatocytes, which are the most common cell type in our liver. And the other group leader had a background more in mathematics and computer science, and at that time he was developing computational tools for the statistical analysis of gene expression microarrays. That was the hot topic at that time. During that time I developed, among others, an R bioconductor software package for the analysis of DNA methylation data, and I have applied this tool for the analysis of DNA methylation data in human embryonic stem cells, and during the early differentiation of these stem cells towards endodermal lineage, which is functional hepatocytes. That study we published in 2010, and that was also the basis for me to graduate the same year. And from there, my journey has led me here to the US, where I continued to analyze DNA methylation in other epigenetic marks in pluripotent stem cells, but also in other normal human cell types. And during the years, I was more and more involved in several genetic and epigenetic studies of normal human cell types, but also looking into cancer tissues. And during that time, I developed a deep interest and some expertise in childhood cancers, or more specifically in childhood brain tumors. And during the last few years, we have published a number of I think, quite important studies in quite high-profiling scientific journals, and this success, I think, has enabled you to get this assistant professor position here at UCSD, and also forms the basis of my ongoing research program here. So I could now, I think, continue for hours to talk about our recent progress in childhood cancers. Just a few examples. We have identified many different types of mutations and structural variants that are very characteristic for different childhood brain tumors. We have also developed an approach to classify brain tumors into clinically relevant brain tumor subgroups and subtypes using epigenetic signatures and machine learning methods. Another important aspect of our work is on our ongoing work is that we use genetic, transcriptomic and epigenetic analysis of brain tumor biopsies to identify personalized treatment options for patients diagnosed here locally at San Diego's Brady Children's Hospital. However, for today I will have to focus on a more specific topic, and as announced, this will be on chromatin states and chromosome conformations. So, what are chromatin states and what are chromosome conformations, and why am I interested in these chromatin states and conformations in the context of cancer? We'll test those. They will work. Chromatin states. So what are chromatin states? Chromatin states are basically a concept to annotate genomes. That is what we're trying to achieve. Another thing that we would like to achieve is we want to assign functions to especially to non-coding DNA. I mean, we know that the genome consists of 3 billion base pairs, and only a minority of the genome actually encodes for genes. So we want to annotate the functions of non-coding DNA. So I think the estimate at the moment is that 3 to 5% of the genome actually encodes for genes. And these chromatin states are defined by epigenetic information or epigenetic marks. And chromatin states are defined not only by one epigenetic marks, but by multiple. Multiple epigenetic marks. So probably the next question is, what are epigenetic marks? That might be a general thing to discuss. Any idea? What is an epigenetic mark? One? Yes. Yes, absolutely. DNA methylation is an epigenetic mark. And actually, we have developed this brain tumor classifier based on DNA methylation alone. But there are other epigenetic marks, for example, histone modifications. Or maybe the accessible chromatin might be also one way to identify epigenetic marks. So in general, epigenetic marks is everything that somehow interacts or alters the DNA without changing the DNA itself. The integrity of the DNA remains the same, but we can add modifications to the DNA, or we can package the DNA, and all of these things are reversible and can be inherited during cell cycle. Those are more or less the definitions of epigenetic marks. For the concept of chromatin states, we mostly use, or it is most common to use histone modifications. In principle, you can use anything, but histone modifications have been most extensively used to define chromatin states. So the next question, probably is what are histones. So histones are protein complexes. And I have to say the biology of histones is pretty complex. There are more than 17 different types of histones, histone variants. More than 70. That's a German way of writing 7. 70 different histone variants. There is a database available. It's called Histone DB, where you can look up all the different histones. So that means really throughout the human genome there are more than 70 different genes that encode for different histone variants. The core histones are called H3, H2B, H3, and H4. And the function of these histones is to package DNA into structural units called the nucleosomes. So it's basically package DNA into nucleosomes. Or another way to look at that is to say, or to make a definition, that DNA that is packaged around these histones, DNA plus histones, defines our chromatin. So whenever we talk about chromatin, we typically think about the DNA packaged, wrapped around histones, packaged, and that concept is on the one hand important for putting the huge amount of DNA into the nucleus, and on the other hand, this is very important for gene regulation.
So what about larger scale structure like loops and extrusion? How does that kind of fit in with the umbrella for?
Right. So loops. I mean, I'm covering that later when I talk about chromatin conformations. Maybe let me postpone the question to that. Yeah. Okay. Thank you. And yeah, I mean, it's also important for gene regulation. Right. Okay. Maybe just schematically show how those look like. So that would be the protein complex formed out of the core histones, H3, H4, H2A, and H2B. And these histones have another interesting characteristic. They all have an amino acid tail. So that means when you look at this protein complex, there is literally a sequence of amino acids, acid tail coming out of these histones. And these tails encode a very important information. So first of all, we just numerate them. We can simply say, well, this is the histone tail. This is the histone tail amino acid position 1, position 2, position 4, and so on. And the amino acids can have different types of modifications. So the DNA can consist in different conformations. We can have tightly packaged DNA, where we have the DNA wrapped tightly around these histones. So when we look at that, the DNA goes around here, goes around here, goes around here, and then we have other regions in the genome where the DNA is not packaged as tightly, where we have these linkers, and we have genes sitting inside or enhancers sitting inside. And typically, these tightly packed chromatin structures are associated with downregulated genes. We don't see a lot of gene regulation here, whereas these open chromatin or accessible chromatin are those regions where we have, in a specific cell type, most of the transcription and gene expression going on. And the interesting thing is that these tails have different marks at these different compartments here. So for example, it is known that the lysine, depicted as K, on position number 27, tends to be acetylated. So there is an acetylation group here at this K27 in all the regions where we have a lot of gene expression. Right. So that would go from H3. So the tail of the protein component here, H3, with the acetylation on the lysine K27 tells us a lot about the activity of this region. Please.
Yeah, for the loose chromatin state, are the genes able to be transcribed because the histone is actually like undone, or is are the relevant enzymes able to go like still onto the DNA while it's still on the histone?
Right. So I mean, this is a complex gene regulatory mechanism that is going on here. There are genes known to be able to bind this packed chromatin and then open up the DNA. Once the DNA is open, other genes and transcription factors can then bind and activate the expression of the genes that sit there. And I mean, all of these mechanisms, we know a couple of genes that are responsible for opening up chromatin, we know others that then bind here, others that repress and package the DNA, but there's still a lot to learn about these mechanisms. And then on the other hand, wherever we have tightly packed chromatin, we also have the lysine in position K27, but in this case, we have a methylation group sitting here. And this is just one specific amino acid sitting on the tail of the H3 component here that tells us already a lot of regions that have active gene expression and repressed gene expression. There are many of these factors, and it can become very complex. And I guess we have still no clear understanding of all the different modifications and their biological function. But nevertheless, there are a couple of those that are very well researched. And the majority of the research out there focuses on 6 of these modifications. The one is the one that I already mentioned. It's the H3k27 acetylation. That is a mark that is clearly associated with active. It's associated with active enhancers and also promoters. And the distinction between the enhancers and promoters is simply that promoters are all the DNA regions that are really very close to the transcription start site of the genes, whereas enhancers can be located far away without being very proximal to any gene, even though they still have a function and regulate the expression of some genes. Another one is the H3k4 ME1. So that is a monomethylation at the lysine on position 4. This is associated with active enhancers. You typically don't see this mark anywhere close to the transcription start site of any genes. Another one is H3k4 ME3, which sits at active promoters. We have a couple of repressive marks, including the H3k27 trimethylation, the one that I mentioned here. This is repressed. chromatin. What we know about this specific histone modification is that there are a specific subset of genes. They all belong to the subgroup of so-called polycom genes that mediate the trimethylation of this histone. We have another repressive mark, H3k27 is also repressed chromatin, but it's not regulated by the polycom genes. And then there is the sixth mark, which is the H3k27 ME3. And this is a mark that we typically see at genes, at gene bodies, transcribed gene bodies. So this mark sits not at the promoter regions like the K27 acetylation or the K4 trimethylation, but it sits downstream of actively transcribed genes along the axons. The question is, how do we measure all of these histone modifications in the cell system or in tissue? And we use chromatin immunoprecipitation as the technique, or shortly it's CHIP. Chromatin immunoprecipitation, and that means we have antibodies that have been designed to specifically recognize histone modifications or histones with specific histone modifications. So we would add antibodies that can recognize these modifications here. And then we would share our DNA into pieces using ultrasound and sonication. So the DNA will be shared into pieces of maybe 200 to 300 base pairs length. And to measure, for example, the K27 acetylation, we would clearly need a different type of antibody. And then we would pull down those DNA fragments that are associated with the one or the other mark, depending on the antibody that we have used. So that would mean so that would mean we get a sample where we have these shared DNA fragments, 200, 300 base pairs, depending on the resolution. And most of those have histones associated with a specific mark. It's an enrichment. And then we typically use a control. So whatever tissue we are analyzing, we separate it in two parts. For one, we apply this antibody, and for the other, we don't. But we do all the DNA sharing and everything else. So we will have a random mixture of DNA fragments here that might have some types of modifications still attached to it, but it's not enriched for a specific mark. So when we then want to identify which regions in the genome are enriched for the one mark or for the other mark, or in other words, to identify those regions that are actively transcribed in the sample, we would then do sequencing of all these little DNA fragments, and then we would end up with a text file full of these DNA sequences that tell us nothing in the first instance, but we have to align them back to the reference genome, and there are plenty of tools available to do that. And then when we look at the genome, and I just depict one example, let's say we are somewhere here, chromosome 1, and let's say that we have a gene that sits here. Here's the transcription start site, and the gene is expressed or not. And we would now get a lot of DNA fragments or sequence DNA fragments that align to this region of the genome. And now we would probably separate the chromosome into smaller bins. And the bin size or the window size somewhat depends on what the resolution of the data is you started with. If you have 200 or 300 base pairs, or maybe larger fragments or smaller fragments. You can do that on a base pair level if you want to, but I don't know, typically we do it on 50 base pair levels. And then you can, or maybe even larger, it really depends. Let's assume 200 base pair windows, and then you would count for each of these windows how many alignments fall into the window, and then you would get a curve that looks maybe something like that. And because we have this control experiment where we don't have any specific enrichment, we would, again, chromosome 1, we would get, I don't know, a couple of reads that fall randomly throughout the genome. There are some regions that are biased towards more reads, so we get some kind of a certain background, and then the challenge is simply to identify those regions that are enriched in the IP, in the chip sample compared to the normal, and then identify these regions marked by one specific histone modification. So that you can do with one chromatin mark in the specific sample, and then you, if you use the K27 acetylation, or antibody specific for the K27 acetylation, you can see that we now identified maybe an enhancer region, because it sits quite far away from the gene, but it still is an active mark, so something must go on here, probably transcription factors bind here, and do some gene regulation. But we can do that not only for one mark, we can do that for several marks, and that has been extensively done during the last couple of years, mostly focusing on these 6 marks. And then it becomes, of course, a little bit more interesting because we have some kind of combinatorics. So let's assume that our gene sits again here. There's the TSS, and now the gene model sits somewhere here. Okay, and then we have now these 6 marks, and we again divide our genome into windows. Let's stick maybe to these 200 base pair windows. I think that is for the definition of the chromatin states one of the most widely used parameters. So we would divide again the entire chromosome throughout the entire genome into these 200 base pair windows. And for each mark, we would then estimate if we have an enrichment over the background or not, and come to a binarized table. So when we look at the K27 acetylation, we probably get a 0 here, we get a 1 in this window here, and we might get another one here. If we still have this enrichment in this example, then we will get a 0 here, a 0 here, and everywhere else. And the K for monomethylation, that is a mark for the enhancer, that would probably also get the 1 here and the 1 here, because that's an enhancer region. The K for trimethylation would not give us an enrichment at distal regulatory elements, but it would probably, if that is the activated gene here, have an enrichment around the promoter. So at a certain point, these windows would become 1s and then 0s again. The K27 trimethylation in this example, because now I'm taking an example of an actively transcribed gene, would probably be 0 throughout the locus. The other repressive mark probably 2. And the H3k27 trimethylation would then, my gene is a little bit shorter, there would probably be 0 here, and then along the gene, I have these ones along the gene body of an actively transcribed gene. There are some states like this one. This would be the quiescent state, because we don't see any signals, at least not in the mark that we have mapped here. Chromatin state. And in a naive approach, or in an approach how probably I would have tackled that, if I were the first one to define chromatin states, I would have now simply looked for all possible combinations that these marks can have. And because we have a binary value here, and because we have 6 different marks, we could have 2 to the power of 664 different chromatin states in theory. But I mean, by doing the entire exercise in human tissues, we find that many of these states simply do not exist. Many combinations do not exist. And there was a lot of back and forth for the field to define how many states there are actually. And there are different models presented. There were different models presented during the last years. And typically, the different models range between 15 and 18 different chromatin states that define distinct functional units throughout the genome. And there's one tool that is most commonly used for that. It's called Chromatin. More space. And that was really the first one and most widely used tool, and there are many more that have been published during the last couple of years. The idea of Chromatin is to learn and to characterize chromatin states, of course. And it integrates different or multiple epigenetic marks. What else? And of course the concept that is used is the multivariate hidden mark of model. And the model is basically the presence and absence of these chromatin marks in a binarized way. There are other methods available that try to be more quantitative to allow for higher numbers if there's a high enrichment. But Chromatin really does this binarization of the data and then tries to learn these chromatin states. So it was mostly by a big consortium called the Roadmap Epigenomics. That was a huge effort, mostly paid by the NIH, and this has eaten millions and millions of dollars. They have, over the years, including us, mapped 90, at least 98. They have mapped 98 normal human cell types and tissues by using this entire pipeline here, looking for these 6 marks. Again, the 18-state model is the most commonly used one, and it has these active states, looking for enhanced promoters. It has repressed states. It has its quiescent state, of course, and it has also bivalent states. Bivalent states are states where we see both an active chromatin mark and a repressed chromatin mark. And typically the genes close to these bivalent states are not expressed, but those are described as being genes poised for expression. That means during the differentiation process, it is very likely that a gene that has a bivalent chromatin state will be expressed maybe a few hours later. Yeah, that's basically the entire concept of chromatin states. And this is the most comprehensive approach to try to annotate our genome-wide. We now know in more than almost 100 cell types where these enhancers are located, where repressive chromatin is, and this is a very valuable reference cohort. 100 cell types sounds a lot, but it is far from being complete, especially when we look into our brains. We have many more different and very specialized cell types. We just need to think about all the efforts to do single cell analysis, and we now know that even within a heterogeneous tissue, we have many different, more specialized cell types. And I think we are far from being complete here and from knowing the entire enhancer and chromatin state landscape of all tissues. But nevertheless, that was a huge effort, and that's more or less the current state of research about annotating our genome. And then the other concept, and I think I have a little time left, is about the chromosome conformation. That was basically the second part. That will be a little bit shorter, and I motivate that here because the question is how, I mean, it's easy to interpret a promoter that is in an active state, because we know that all the transcription factors that bind here in the promoter region then regulate and activate the gene, and it's easy to identify these transcription factors that activate this gene. It is more complicated when we look at these enhancer regions. So we can have these cases where, again, let's say we look at chromosome 1, we have a gene located here, and now we have done the entire exercise here and we identify, let's say, an enhancer element that is strongly marked by the H3k27 Acetylation. And the question is this enhancer actually regulating this gene or not? Especially because it is very likely that there might be another gene located here. And the question really is this an enhancer that regulates this gene, or is it an enhancer that regulates this gene? So this is why we are very interested in mapping the 3D conformation of these genomes in different cell types. And we would like to figure out how is the DNA structure, and that probably goes back to your question when we look at these DNA loops. So I mean, the DNA is highly packed not only around the histones, but they also form these loops here. Then there are spacers again, and then the DNA is packed into these loops again. And there might be another spacer here. And the chromatin, or the DNA is wrapped around these chromatins within this substructure. And I would imagine that goes towards your question. Right? So we have these larger loops and compartments of the DNA, but they can be within these loops tightly packed or more open. So when we now map the DNA interaction, we can see that there are these compartments coming up. Like this one, or like this one is coming up, or this compartment. And we now know that there is a lot of DNA interactions between DNA stretches on this compartment, whereas there is very little DNA interaction between these compartments. So that means here can be an enhancer, and here can be the target gene. And even though when we look at the DNA as a linear stretch, this enhancer can be located millions of base pairs away from its actual target gene. But by this formation of the DNA loop, the enhancer is being brought into the vicinity of the promoter region of its target gene, and then maybe even skips a lot of genes that sit along the stretch, and this enhancer does not regulate the expression of these genes at all. Yeah, it is still a very important research topic to identify these enhancer gene interaction networks. And it might be important to understand what transcription factors actually regulate the gene. It might be important to understand in the context of cancer that we might have a cancer where we don't find any gene that has a significant mutation, but we might find an enhancer that has a mutation here. There are not many, but rare cases reported so far that there can be some mutational processes going on that affect enhancers and not genes. And by looking only at genes, we are blind to that. By including the non-coding DNA, we might find cases of mutated enhancers, and by mapping the 3D genome organization, we get a better idea of the genes that are actually regulated by these enhancers, or then maybe deregulated due to the mutational status. So, how to analyze these 3D conformations. There's one technology that is widely used, it's called HiC. And that's an approach that tries to identify the 3D genome conformation of a given sample or tissue type, genome-wide. Without going into the details how all the biochemics and the assays actually work, I just want to depict how the data typically looks like when we have processed and aligned and sequenced everything. So let's again look at chromosome 1. And now we put chromosome 1 here onto the y-axis, and then the chromosome would align along the diagonal here, and we would have our gene sitting here, or when we map it, the diagonal, the gene would be here, and we might have an enhancer here. And then we do all these DNA interaction analysis, and then we would get a signal of a region sitting here that is in contact with the DNA region that sits here, and that would give us an interaction signal sitting somewhere here, and then we know that there are these compartments here and that there is a lot of interaction within these compartments, and then we would see, and because it's a mirror matrix, we would get such a box that represents basically many interactions within such a compartment, and then we would see no interactions here, and then we would see a lot of interactions between DNA fragments going on here, and then we might see another box over here. And I mean, that was not known a couple of years before, but when these assays have been developed and cells and tissues have been processed, suddenly these patterns arose, and then it was becoming very clear that the DNA is organized in these compartments, or how they were termed so-called TADs, topologically associated domains. These TAD domains can be very stable. It has been estimated that 70% of those seem to be very stable in different cell types, but they can also shift around, and the formation of new TADs can lead to interactions between an enhancer that sits maybe here and then activates a gene in a TAD domain that was not accessible in a different cell type. Again, as I said, there have been some reports in cancer that there are mutations or structural variants going on that disturb these structures and then lead to disturb gene regulation simply because an enhancer that should be separated from some genes in another TAD suddenly activates the gene it should not. This is some mechanism that has been coined the enhancer hijacking. And that basically brings me to the last topic I just briefly would like to discuss. Why are chromatin states and conformations important in cancer? I mean, I already mentioned a couple of those aspects in my case specifically. Why are these chromatin states and conformations important for brain tumors? And I would like to explain that a little bit further. So there's one brain tumor type we are very interested in. They are called megaloblastomas. Or short. So megaloblastomas are brain tumors that arise in the cerebellum. So when we look at the brain, let's say this is the brain stem, and we have the cerebellum sitting here, and then we have the brain here. So this is the posterior fossa region of our brain, and here sits our cerebellum. And megaloblastomas typically arise in the cerebellum of the brain. So megaloblastomas are a disease of the cerebellum. There are other tumor types that arise in the cerebellum, but megaloblastomas are clearly the most frequent brain tumor types, or childhood brain tumor types. The standard of care includes maximal surgery. So that is always the best thing to remove as much of the tumor as possible. The brain is a difficult organ, as you know. And then this is followed by radiation and chemotherapy to kill the remainder of the tumor that couldn't be removed. The survival rates are pretty okay. They are at 70 to 80%. If the tumor has not spread, the survival rates drop to less than 60% if there are metastases. And the long-term goal of our brain tumor research, including the research that we do in my lab, is basically threefold. I mean, first of all, it's really to understand the basic biology here, to understand more about tumor biology. That is, I think, the number one thing that we do. Simply trying to understand what is going on here in terms of gene expression, in terms of epigenetic regulation, in terms of genetic mutations or structural variants and rearrangement. Then it is about doing clinical research. That is basically everything that involves testing of mechanisms in living system, typically in cell lines, but also in model organisms like mouse or mice. So in vivo, in vitro research, whenever we have identified, let's say, a gene affected by a very tumor-specific rearrangement or by a 3D genome confirmation that seems to be specific to a tumor, one could try to target these genes in medulloblastomar cell lines to see if by targeting or inhibiting this specific gene we see a decrease of the cell survival in vitro or in mouse models. And the third thing then are clinical trials. So whenever there is a target that seems to be effective throughout different models, and if we can find a drug that has been approved by the FDA and is known to inhibit the function of a specific tumor dependency gene, it would then be tested in clinical trials at selected cohorts of medulloblastoma patients, for example. And we typically cover mostly this part. We also work very closely with clinicians here at the UCSD and are in very frequent interactions with each other, trying to identify targets, then going back to the more experimental labs and doing further preclinical research. And my lab is not so much involved in these clinical trials. This is something that really happens then in the cancer center, either at the Reagan Children's or at the Moores. Last thing, because I think I'm running out of time now. It has been observed by intensively sequencing hundreds and hundreds of these medulloblastomas that there are a couple of genes that are recurrently mutated. And that there is a group of genes that appear to be affected again and again. One of them is the KMT2 genes. There is a KMT2C, a KMT2D gene, which again we find mutated in maybe 20 or 30% of specific subgroups of these medulloblastomas. There are other genes like the KDM6A or the PRDM6. And the interesting thing is that by identifying more and more genes that are mutated in medulloblastoma, we find that these genes all have something in common, or many of these genes have something in common. They are all regulators of the chromatin modifier. So this gene is, the name of the gene is the lysine. The K comes from the histone amino acid on the tail of the H3, which is lysine methyltransferase, and then one gene of this gene family. So we know that this gene is involved in adding monomethylation, single methylations to the K27 of the H3 tail. Right? And the same thing is here. We don't know a lot about many of these genes. We just know that they are somehow involved in modifying the chromatin. And then the research question is, of course, if we have these mutations in these many chromatin modifiers, how is the effect on the chromatin states? How is the effect on the 3D genome conformation? So what we are doing right now is we are mapping all these different marks that have been mapped in many different normal human cell types. We do map those in a cohort of medulloblastoma tumors that we have assembled. And we are looking for enhancers, but we are also learning these states in medulloblastomas. And what we observe is that we do identify a couple of states, chromatin states, that have not been observed in normal human tissues, and we try to associate that back, or we think that is due to mutations in these chromatin modifiers. And now we try to identify exactly the regions in the genome where we have these tumor-specific chromatin states. We do this 3D genome organization or 3D genome analysis, try to identify the target genes, and we do find a couple of genes, maybe something like 100, 150 genes that seem to be specifically deregulated by mutations in these genes, and that again helps us to identify those genes that might be the best targets to inhibit or to disturb somehow, to interfere with the proliferation and the growth of these tumors. And with that, I stop. Thank you very much for your attention. I hope you have a broad overview of what we are doing in the lab.
Yeah, well, thank you so much for coming out. Yeah, do you guys
have any questions?
Yeah? Oh, sure, of course. Please.
I have two questions if I could. The first is that it seemed like with the hidden Markov model, like Chrome-HMM, that the major bottleneck was that you actually have to perform all those ChIP-seq experiments, get the data, feed it to the Markov model. Is that correct? That's kind of...
That's correct.
Okay. Has anyone done any work trying to infer chromatin states from sequences? And if so, has it been successful at all?
Yes, indeed. So while mapping all of these 100 different cell types, it turned out that in a couple of these experiments or tissue types, individual ChIP sequencing experiments didn't work. So one approach would be to simply take a similar sample and do the same thing. But there was a very interesting paper that tried to infer missing chromatin states. So when you have this model, and then you know which chromatin states, which combinations exist, especially the Chrome-HMM, the hidden Markov model, not only looks at the states, but it also looks at the transition between the states. So you can, with a certain reliability, actually predict states when you have missing data. Predicting it from the DNA sequence alone, there's probably some research done on that. I wouldn't be surprised, and you can probably do that, but the problem is always that you can always achieve a certain reliability, a certain degree of quality, but it will never be 100%. And I'm telling you that when we enroll a patient and do the chromatin state analysis, we cannot say with high probability that this enhancer we really need to know. So typically try to get a most complete data set. But it's a very interesting research area.
Yeah.
Yes?
I had two questions, one piggybacking off of his. So, so going off of what he was saying, wouldn't there be able to be some kind of like machine learning mechanism to like possibly compare healthy genomes versus like these cells where there is methylation, modification, stuff like that, and possibly look into associations between methylation and the actual sequence of the genome?
Yeah, absolutely. Now as I'm thinking more about that, I think there have been several methods published that do exactly that. I don't recall how good they work actually.
Sure.
Yeah, but I mean, if you can come up with such a method, we could save. I mean, when you can come up and prove that this method is really waterproof, we can save a lot of money, and we can simply annotate that. But I mean, the DNA sequence is fixed. Let's not forget about that. Yeah, the DNA sequence itself will. I mean, except we have mutation. That might be an interesting research direction. How does a mutation in the DNA sequence affect the chromatin state?
Well, I was thinking more in terms of like mRNA expression or non-coding sequence or non-coding mRNAs, stuff like that.
Right, but all of that, the mRNA expression, all the histone modifications or other epigenetic marks are very dynamic. They can be very different in different cell types. If I look into my hepatocytes or in my big toe or whatever, there will never be the same genes or the same set of genes expressed, and there will not always be the same chromatin state. So even though the DNA sequence is given, there is a lot of epigenetic dynamics going on. And without any additional information, it will probably not be possible to just calculate the chromatin state just by looking in a specific cell type, by looking only in the DNA. It would be more like, well, let's map only 2 marks, or maybe, and then we can infer all the other marks or something.
Sure.
In my best case.
Yeah, and then my second question might be just really stupid, but for like treatment modalities, would you only be able to target those genes that code for the acetylation and methylation marks, or could you actually tag the marks that are already present on the cells, or and possibly tag them for for other therapies like chemo, immune therapy? I'm not sure if the presence of the histone definitely indicates healthy cell, or if it's like a histone could indicate a healthy cell or a cancerous cell.
Mm-hmm. Right. I mean, what we do observe is that in these tumors we find enhancers or chromatin states that we haven't seen before. One reason could be that this is a tumor-specific event, that because of the mutation in the gene, there is an enhancer or something forming that wasn't there before. Another explanation could be that during the development of our brain, I mean, that is a very complex process from the first zygote up to a fully adult functional human brain. There are many different cell types. I mean, at the beginning I said our adult brain has many different cell types that we probably have not mapped yet, but I could push that even further and say maybe during the entire development of the brain there might be even more intermediate cell types that typically don't exist in an adult brain anymore, and we know that there are some of these cell types. So it is also very, because we are looking into childhood brain tumors here, so many of these tumors actually develop during the embryonic state. So what we are looking here might be enhancers that are not necessarily only specific to a cancer, but that might be active during an early differentiation state, and then the cancer develops because of the genetic hit, and then grows out into a tumor and maintains this epigenetic mark. I think I'm not really answering your questions. I think you asked more about can we target these epigenetic marks that we observed in these tumors, and that is very, very difficult. So all the clinical trials or all the efforts to come up with more targeted therapies, and not only do radiation and surgery, but really develop a very targeted therapy approach, really still focus on targeting genes, because genes are being expressed and there's a way to silence them. And these chromatin modifiers are unfortunately very bad targets. Even though we know that there is a mutation, it is very difficult to develop a drug that targets these genes. There are a couple of characteristics, good targets. I don't know, it's not really my area, but if you talk to pharma companies, they always tell you they are looking for very specific genes that are expressed in the cell surface, that are more accessible to drugs and inhibitors. So the idea is to find tumor-specific or maybe cell of origin specific enhancers to identify their target genes by doing this 3D genome analysis, and then hope to identify good drug targets that are upregulated in this specific gene compared to normal.
Thank you.
That's the idea.